Hey everyone, welcome back to The Explainer. Today, we're taking a journey into the hidden layers of genetics. We're going to uncover a secret RNA code that is completely changing how we understand molecular biology. Get ready, because we're doing a deep dive into the fascinating world of RNA modifications, and we'll be revealing the invisible systems that literally tell our cells exactly how to function.
Okay, let's dive right into this. Here's our roadmap for today. We'll start with the hidden RNA code, look at how we decode M6A with MeRIP, walk through the MeRIP sequencing workflow, check out a really cool case study on tomato ripening, and wrap up with CD genomics sequencing solutions.
Section 1. The hidden RNA code, beyond the basic genetic blueprint.
You know that genetic code we all learn in school, the standard A, C, T, and G of DNA, and how they transcribe into messenger RNA? Well, turns out that's really only part of the story. There's this whole other layer of dynamic regulation happening right on top of those basic blueprints, and it completely transforms how that code behaves. To really understand this, we need to talk about N6-methyladenosine, or M6A for short. This is the absolute most prevalent form of methylation modification found in the mRNA sequences of eukaryotes. Simply put, it's the most common way our cells chemically modify mRNA to functionally regulate what the transcriptome is doing at any given moment.
I absolutely love this analogy because it makes a super complex molecular concept instantly click. Think of it like this. If messenger RNA is the text of a printed book, M6A is like a bright highlighter marking specific sentences, telling the cell exactly how to read and use that specific information. It doesn't actually change the words, the sequence is exactly the same, but it completely changes what the cell focuses on and what it actually does with that biological text.
And where does this highlighter actually matter? Well, virtually everywhere. This tiny little modification influences incredibly foundational biological processes. We're talking about mRNA splicing, exporting from the nucleus, localization, translation efficiency, and overall mRNA stability. And if we zoom out to the organism level, it's a major player in stem cell differentiation, keeping our circadian rhythms in check, and even managing how organisms respond to environmental stress. It's a really big deal.
Section 2. Decoding M6A with MeRIP. Capturing the highlighted text.
So now that we know this highlighted text exists and does so much heavy lifting, how do we actually read these hidden molecular highlights in the lab? The answer is our main investigative tool, MeRIP-seq. That stands for Methylated RNA Immunoprecipitation Sequencing. Let's break that down a bit. It's a really elegant technique that uses M6A-specific antibodies to physically capture and then sequence only the enriched, modified RNA fragments. We're essentially going fishing, but specifically for that highlighted text.
But if you're actually running this in a lab, there's a really essential methodological question that always pops up. Why is it absolutely necessary to simultaneously measure both input and IP samples in your MeRIP experiments? Why run two parallel tracks just to answer one question?
And this brilliantly illustrates the whole mystery-solving nature of the experiment. The IP sample is where we use that M6A antibody to specifically capture the methylated RNA. That is our true signal. On the flip side, the input sample is just all the fragmented RNA straight from the source, absolutely no antibody enrichment. It acts as our baseline control. By comparing the two, bioinformatics can basically subtract the background noise found in the input from the IP sample, maximizing the precision of our actual M6A signal.
Section 3. The MeRIP sequencing workflow, from raw sample to digital data.
With that foundational concept in place, let's look at the actual laboratory mechanics. I promised you a step-by-step look at how this highly specialized sequencing is executed, going all the way from a raw biological sample to digital genomic data. Here's how the workflow unfolds. First up, poly-A-selected RNA is fragmented into tiny pieces, exactly 100 nucleotides long. Second, the IP sample goes through immunoprecipitation using an anti-M6A affinity-purified antibody. Third, both those IP and input samples are subjected to Illumina next-generation sequencing. Since the M6A can be anywhere along the fragments, we get multiple overlapping reads. And finally, powerful bioinformatics step in, using peak detection algorithms to accurately predict exactly where those M6A sites live.
Now, like any advanced laboratory tool, this methodology has a very specific profile. Its strengths are absolutely profound. It gives you high-throughput analysis, incredible sensitivity for low-abundance modifications, and exact spatial resolution. But you have to be precise. The catch? is that it absolutely requires exceptionally high-quality antibodies to work properly, and it heavily relies on highly specialized bioinformatics to untangle the incredibly complex data it spits out.
Section 4. Case Study. Tomato Ripening. Observing m6A in action.
Let's move on and see how this builds into a tangible scenario. We're going to apply this exact workflow to a really fascinating, non-clinical, real-world example in plant biology to see m6A in action. Take a look at this landmark 2019 study published in Genome Biology. And yes, fun fact, while a tomato is culinarily a vegetable, biologically it's a fruit. These researchers successfully utilized MeRIP-seq to map RNA methylomes to understand exactly how these fruits physically change and mature during the ripening process at a molecular level. Their main focus was uncovering the m6A-mediated regulation of a DNA demethylase gene known as SLDML2.
To get their answers, the researchers followed a really strict library construction phase. They extracted the total RNA directly from the tomato samples, enriched the specific RNA they needed, fragmented that RNA down, and meticulously constructed the precise MeRIP-seq libraries required for sequencing.
And get this, what they found was visually and biologically stunning. They discovered that RNA methylation is incredibly dynamic. As the tomato fruit ripens, the overall m6A level gradually decreases. But what's truly mind-blowing is that this decrease perfectly mirrors the patterns of DNA methylation happening at the exact same time.
So, the crucial point is captured beautifully right here in the study's own conclusion. This study preliminarily confirms the connection between DNA methylation and RNA methylation, revealing a new mechanism of fruit ripening regulation. It essentially proves that our m6A highlighter interacts directly with the master DNA blueprint itself. How cool is that? Section 5. CD Genomics Sequencing Solutions, your premier partner for MeRIP-seq.
Zooming out from our tomato study, you might be asking yourself how you can reliably execute these complex multi-omics projects for your own research. Well, this brings us to CD Genomics. They are an optimal professional partner for executing precise MeRIP-seq analysis, and they offer massive technical advantages for your benchwork. We're talking high flexibility across literally any species you're studying. They deliver incredible precision, locating binding sites within a super tight range of 100 to 200 nucleotides. And most importantly, they offer a true digital signal. This completely eliminates the cross-reactivity and analog background noise you get with older chip hybridization methods. Add all that to a comprehensive one-stop platform, flexible library construction, and exceptional experimental reproducibility, and you've got a powerhouse resource on your hands.
Now, what's really interesting here is looking at their true end-to-end service. The rigorous standards immediately stand out. It starts with strict sample quality control, like requiring a RIN value of 7.0 or higher. Then it moves through flexible sequencing options on world-class platforms like the Illumina HiSeq X and NextSeq 500. And it wraps up with comprehensive bioinformatics deliverables, handing you publication-ready data like peak identification, transcript distribution, and rigorous differential peak analysis. Which leaves us right here. If the hidden highlighter of m6A dynamically controls everything from a simple tomato's ripeness to complex stem cell differentiation, what other biological mysteries could your own samples reveal with MeRIP sequencing? Imagine the potential of your next big project when you're armed with the right tools from CD Genomics. Keep asking big questions, keep looking for those hidden molecular highlights, and until next time, stay focused and stay curious.